In this module, we will discuss some of the in vitro assays to quantify the whether a cell, when they adhere on a material substrate, whether the cell are kind of viable, they are like metabolically active cells, and or whether the cell is already dead. That is number one. Number two is that whether after adhering on a material substrate, whether cell is changing its functionality. Remember, I am showing here the two different cells with different nucleus being shaded differently, just to show that they are show that it is a differentiated cell. So, it is a more matured cell. Now, the first one, which is most widely used in the biomaterials community, is that MTT assay. So, if one can go through most of the published papers in the field of biomaterials, they would notice that this MTT assay is most widely used as one of the biochemical assay for determination of the to quantify the cell viability, and subsequently if it is measured at different time point, then it can also give an idea about the cell growth on a material substrate. So, before I start discussing on this any biochemical assays, let me tell you that the fundamental principle of any biochemical assay is that you use certain reagent which will essentially interact with some of the cellular organelles. It can be mitochondria, or it can be it can intercalate with DNA of the cell of the cells and so on. And once it interacts with these some of the cellular organelles or some biomolecules or some biological macromolecules within the cell, then what will happen? It will change the color of that particular organelles or that specific organelles, in case the assay is a colorimetric assay. That means you need to change, you need to determine that what is the change in the optical density before you add that specific biochemical reagent, and after it is being added to the a culture medium. So, I repeat, whenever you use certain biochemical assays, it interacts with some of the organelles within the cell or some biological macromolecules or protein or protein molecules, proteins in the cell structure, and after it interacts with that or specific organelles or biological macromolecules, then what it will do? It will change the color, and then change in the optical density is recorded by some microplate reader or so on, and that optical density, change in optical density will reflect on certain aspect, whether it is a cell viability or whether it is a cell death, something they will something will be detected. So, now in the case of this MTT assay, as I said, it is a colorimetric assay. That means it is based on this color change. So, this is the yellow MTT, and it has a very large expanded chemical formula. It is reduced to purple formagel once it interacts with the mitochondria of the cell. So, essentially MTT assay, this MTT reagent interacts with the mitochondria of the live cells or living cells, and then the process of mitochondrial dehydrogenase takes place, and then formagen crystal forms. Now, formagen crystals has a purple color. So, certainly this purple color this optical density now would be quantified in terms of the increase in absorbance, and this increase in absorbance will be proportional to the number of mitochondrially viable cells. So, therefore, MTT assay essentially determines mitochondrially viable cells or mitochondrially active cells or in one word it actually determines the number of viable cells in solution. So, this is that MTT reagent formula. After the mitochondrial reductase, then it forms this formagen crystals. These formagen crystals in purple in color, and it is shown that this is the live cells. Then after this MTT reacts, then it forms these purple colored formagen crystals, and their absorbance is kind of recorded. So, this is the protocol in a schematic. So, what normally people do, that you can take some particle eluates or some particle eluates, or you can use the bulk samples. Now, this is typically shows this typical culture plate well, like you can use 8 well plates, or you can use 96 well plates and so on. So, you put this so first you have to add this yellow MTT reagent to the cell suspension containing the particles or containing these bulk materials. Now, once it is reduced, then you can extract this one and then put it in the extracted solution, you put it into the 96 well plate, and then you can have some blank one or some control sample, and then you can have this is that actually test sample. This absorbance of the purple colored solution then further is quantified by measuring this absorbance is particular range, that is 500 to 600 nanometer wavelength by a spectrophotometer. Now, once it measures that will give you that what is the number of what would be the relative number of these mitochondrially viable cells. Now, in cell biology often people use the complementary assays. Now, one of the examples of this complementary assays is this lactate dehydrogenase assay, that is the LDH assay. So, what LDH actually does, it quantifies the number of dead cells. Now, one of the things I think emphasized in one of the earlier modules, that in all these biological assays you have to state whatever this biological assay actually is meant for. One should not overstretch it, one should not project it to a large extent. For example, I repeat it, that if that X percentage cells are mitochondrially active or viable, then one cannot simply write that 100 minus X cells are dead. They may be dead, they may not be dead, because 100 minus X cells, out of that certain fraction is dead or certain fraction is in different state of their cell fate processes. So, this LDH it is a marker for the dead cells, and it is released in the culture medium once cell dies due to the damage of the plasma membrane. And the increase in therefore, the LDH activity in culture supernatant is proportional to the number of lysed cells. So, this is a typical rationale of using that LDH assay, and this is LDH is typically a cytosolic enzyme which is present in most eukaryotic cells. So, once it is coming out, that means your cell lysis has taken place, and that is why that LDH is coming out from the cytosol to the extracellular region. So, this is how this schematic looks like. So, you have a damaged cells, that means that cell membrane lost its integrity. As a result, this LDH comes out, and then it forms again the formagen crystal. Now, this is again a highly colored supernatant, and soluble formagen crystals can be measured at similar wavelength region, like 490 nanometer spectrophotometrically, and this will aid in the quantification of the number of dead cells. Third one is that picogreen assay. Picogreen is actually a fluorescent nucleic acid. So, it is a very ultra sensitive fluorescent nucleic acid stain for quantifying double stranded DNA. So, remember this is only used for the double stranded DNA. It is very simpler, and only a fraction of the sample is needed, like it is in the order of 25 picogram per milliliter, and single concentration of picogreen reagent allows detection over the full dynamic range of the assay. and so this is the typical protocol. So one is to first prepare the working solution of picogreen reagent. After that similar to other assays, that one is to dilute with that unknown sample of a desired volume, then add equal volume of the working solutions and measure the sample fluorescence in the fluorometer. So this is how this picogreen assay that works. So let me summarize these 3 different assays that I have just discussed in last 10 minutes or so. So first one is the MTT assay, which is used to measure the metabolically active cells, and then if that metabolically active cells is measured at 3 or 4 different time points in culture like T1, T2, T3, T4, or in other words let us say 1 day, 3 day, 5 days, 7 days, then if you can plot this one, that how these cell numbers is increasing, then you can see that how the cell growth takes place. That kind of quantification is possible. When metabolic events leads to necrosis, the reduction in cell viability, this assay is based on mitochondrial respiratory activity and would give early signs of toxicity. LDH assay, it is a non-destructive again measurement technique, and this increase in the LDH activity in solution essentially is a measure of the cell lysis. Picogreen assay, it is just to quantify the double stranded DNA and is highly selective for double stranded DNA over single stranded DNA and RNA. Now some practical guidelines, I mean if one does experiments in cell biology, particularly in the context of biomaterials research. So the time points of these assays or the culture period should be based on the doubling time of cells. Now some of the doubling time of cells has been mentioned before, but on the basis of the doubling time, let us say if one is to study the proliferation of the C2C12 mouse myoblast cells, then a time point of typical time point 24, 48, 72 hours is fine, because they are typically the C2C12 cells, their doubling time is 12 to 19 hours. And one is to always do complementary assays to avoid any false positive results. For example, presence of bacterial contamination gives higher absorbance values due to reducing environment created by the bacteria. Now another thing that is quite important to mention, like one can use these MTT assays or LDH assays. So particularly the MTT assay is not useful for carbon based materials. Now if you use these carbon nanotube based materials or any other carbon based materials, for example graphene based materials, you want to understand their toxicity towards specific cell lines, one cannot use that MTT assay. So for that you need to use certain other specific assays. So because that MTT reagent interacts with carbon and therefore it gives a false reading which may not reflect the mitochondrially active cells in that particular solution. So as long as these biological reagents or biochemical reagents they do not interact with the materials per se, then you can use, but once it starts reacting with the materials, also material composition, then one has to be very careful. Now for carbon based materials there is another assay, what is called WST1 assay, and this WST1 assay can be used, and the WST1 assay actually provides reliable measure of the mitochondrially active cells on the carbon based materials. The third thing that I have mentioned is that appropriate controls like both positive and negative controls should be used to account for such false positives. Now many times in that biomaterials people use the tissue culture polystyrene or tissue culture plastic cover slip as one of the control where cells normally grow. Now another positive control one can use is like hydroxyapatite as such in case of the bioceramic materials, because hydroxyapatite is known to be the most biocompatible and bioactive material, and therefore different cell types can grow very easily on hydroxyapatite based materials. And what about negative control? Negative control often people use, like they simply use a culture solution and they add H2O2, and if you use that hydrogen peroxide, then normally it kills all the cells, and this H2O2 added cells also can be used as a negative control. Now, choose appropriate assays in such a way that the end product does not interfere with the material or compound tested. This is the point that I just explained you with some examples of the carbon based materials, right. Then this another example is that LDH enzyme released from lyse cells can be inhibited by the material or compound tested, and thereby having less optical density values or lower cytotoxicity. So, although most of the cells are lysed, but the origin of this LDH assay is that LDH is an enzyme which is present in the cytoplasm or cytosol that has to be coming out of the cells to the medium, and unless that LDH in an appropriate amount is released into the medium, you do not have any way to measure their optical density values. So, any compound which is present in the culture medium or whose toxicity you want to assess, if that compound inhibits inhibits the release of the LDH enzyme to the solution, then automatically you can get lower optical density values. Okay. So, other points that I have mentioned, that all biological assays are prone to high variability, hence always conduct independent experiments multiple times and on multiple samples, as well as perform SPSS analysis to get statistically significant data. Because if you do some cell culture experiments today on the same material, and if you do it tomorrow, because since cells are continuously growing in the medium and you are culturing those cells on another non-living substrate, therefore, you may get some variable cell culture variable results from different biological assays. So, definitely one would have to use these assays multiple times and multiple samples and use statistical analysis, which is very very important in the context of cell biology research, because one has to do this appropriate statistical tool so that they can convince others that yes, indeed that statistically significant difference can be obtained as far as the cell functionality is concerned. The other things I have mentioned already, that tissue culture polystyrene or cover slip is used as the most suitable control in cytocompatibility. Now, coming to the differentiation assays. So, differentiation means the differential gene expression, which I have mentioned earlier, and one of the, in the context of the bone tissue engineering applications or orthopedic applications, what is the theme of this course? There are two assays which are important. One is that alkaline phosphatase assay. So, alkaline phosphatase assay is used to examine cell differentiation such as osteogenesis, like as I said that it is for the bone cells, and it is associated with increased expression of ALP on the outer surface of the osteoblast plasma membrane. Essentially, this ALP is expressed more heavily, more at a higher level at the outer surface of the plasma membrane of the osteoblast. And changes in ALP activity also involved in the various physiological and pathological events like bone development or bone related diseases or inflammatory bowel diseases or drug toxicity. Third one, that ALP specific activity is used for the osteoblastic phenotype, and this is important in determining bone mineralization. And fourth one, it is an early stage differentiation. So, for example, if you grow human mesenchymal stem cells, which I will cover in one of the module, and then you are following that osteogenesis lineage of the human mesenchymal stem cells, and then ultimately it will go to osteoblast and subsequently osteocytes. So, all these differentiation aspect needs to you need to grow these human mesenchymal stem cells on certain osteoinductive materials for let us say 2 weeks, then after that you go for the 4 to 6 weeks and so on. So, you can see that at different time frame you are essentially detecting that whether there is any signature of the differentiation of human mesenchymal stem cells to osteoblast. So, that early stage means let us say up to 2 weeks to 3 weeks, you can see whether ALP expression is increased or not. If ALP expression is increased at the early stage, then you can be very sure that this human mesenchymal stem cells is undergoing osteogenesis. Now, after the late after this osteoblast cells from pre-osteoblast, mature osteoblast and finally osteocytes, then you see that expression of the osteocalcin, which is essentially late differentiation marker. So, essentially the progress of differentiation is to be assessed by 2 or 3 different markers, and these complementary assays will give you some quantification that how this differentiation process is taking place at different time points in culture. So, this is little bit more details of this ALP assay. So, you have this PMPP, that is a p nitrophenyl phosphate, and then once you add this alkaline under this alkaline conditions, that p nitrophenyl will undergo transformation to p nitrophenolate, and this you can see that there is a certain color change. And this is again a colorimetric assay. So, it is essentially based on the determination of the optical density change, and this optical density change is essentially monitored by this PNPP activity. Now, upon dephosphorylation, this PNPP essentially turns yellow. So, like that you remember that in case of MTT, your formaggine crystal that turns purple colored, and then purple colored is measured at certain wavelength using spectrophotometrically. So, similarly during the ALP assay, upon dephosphorylation, this PNPP turns yellow, and this is measured at 405 nanometer absorbance. And this assay can detect, this is very sensitive assay, this assay can detect 0.2 nanogram per milliliter active alkaline phosphatase. So, even if this alkaline phosphatase is present in very small amount, ALP assay can pick up this small amount of this alkaline phosphatase, and then it can quantify. So, this is little bit more details on this, how this assay is conducted. So, you first prepare the working solution like as I discussed mentioned for other assays. Then add at least 50 microliter per well biological samples containing alkaline phosphatase. Then you add 50 microliters PNPP as I mentioned before in the last slide. Then mix the reagents and incubate desired temperature for at desired temperature means 37 degrees Celsius for 30 to 60 minutes, roughly up to 1 hour. Then 50 microliter of the stop solution is added, and finally this absorbance is measured at 405 nanometer. So, from this simple description of the different assays, what you can notice that there are 3 or 4 distinctive things that you have to understand, that each assay depends on certain particularly this colorimetric assay depends on the final measurement of the optical density changes at a specific wavelength. Second thing is that you have to understand that what is the sensitivity of these assays, and third thing is that for what kind of purposes that you are essentially measuring that, and what is the origin for all these assays, like whether it interacts with a specific organelles or whether certain enzyme is kind of leached out of that particular cytoplasm of a cell and so on. So, once you know the sufficient background of the each and every assay, then only you will be in a position to analyze and interpret the different values that is coming out of all these assays. So, I will continue the differentiation assay in the next module and complete this part.